Okay, so it's 10:45. We're going to resume the program. So our next speaker is Dr. Terry Reese. He's a senior product manager of cell health at Promega Corporation. Dr. Reese has served as an editor of the in vitro cell-based assays section of the AGM for years now, and we're very delighted to have him talk to us today. So his talk is entitled Treating Cells as Reagents to Design Reproducible Assays. Welcome, Terry, and the floor is yours.
Thank you, Serene. Hopefully this will come up. Okay, thank you for the introduction. And as I go through this, well, first of all, I should also say it's a pleasure to be able to do this. And for those of you who I haven't seen, my colleagues in the AGM, I think since last SLAS, I'll wait until the laughter subsides about the length of my hair and then start right in. So a general outline of what I want to cover today is talking about variability in cell-based assays. And part of what I'll try to convey is to try to get people that use cell-based assays to understand the nature of cells in vitro, talk about phenotypic drift and the importance of establishing standard procedures for handling and culturing cells, talk a little bit about cryopreservation and cell line authentication. As you see here, let me my pointer up. Cell line authentication is in all capital letters, so it must be important. And also mention confirming cell number as a normalizing control. So what causes variability? And the point I want to make is if you understand what causes variability in cell-based assays, then you can do something about it. And you can use the information about the variability to do something about it and improve the assay. Another point I want to make is that most variability is due to the cells themselves and not the results of having variability in the detection reagents. Earlier it was mentioned about having a common batch or a common lot of reagent to do a screen. That's very important, but if there is batch-to-batch variability in the detection reagents, you can often correct for that with the appropriate controls. However, there are situations where there are undefined components in what you're using, like in the medium, for instance, and if it's serum or matrigel that you're using, some of those differences can lead to causing differentiation in the cells and cause changes that are more difficult to deal with. So how can reproducibility be improved? And take-home message right off the bat is to create and follow a standard operating procedure for good cell culture practices and follow that consistently. And that will help you deal with reproducibility problems. Another thing to help you understand the situation is to simply understand the nature of cells in vitro. And what do I mean by that? So I copied down these quotes from presentations over the years, and you can see that some people during a presentation will say that cells have a strategy to develop drug resistance, or cancer wants to suppress the immune system, and then talking about cells as cancer cells learn how they are being attacked, and neurons are happy in the presence of extracellular matrix. So if you think about this, what's wrong with these statements? And they're applying some human characteristic to the cells themselves. Talking about a strategy or wants or learns or feeling happy. Individual cells don't learn. They don't strategize. They don't have wants. They don't feel happy in most cases. Sometimes you'll find a happy cell, okay? But what I try to say is because cells don't think, you have to do the thinking for them. In other words, you have to understand, what the nature of cells in culture really is. So the problem with variability is that cultured cells can change over time in culture, and that those changes can lead to assay variability. One of the major issues with changes that happen in a cell population is phenotypic drift. And so why do cultured cells change or evolve over time? As I say, as it turns out, Darwin was right. There is variation among individual cells in a population, and survival is going to be driven by the fittest, or those cells that proliferate the fastest. And any growth advantage of a subpopulation of cells will eventually become predominant and overtake that population. Okay, so that's something you have to be concerned about. And one of the issues that you have to deal with is a selection pressure that you apply to that population of cells during the subculture process. And that selection pressure, or selecting for a subtype, can certainly lead to phenotypic drift in that population of cells. So what are some of the contributors to phenotypic drift? During the early days of cell culture, the issue was just getting the cells to survive and having enough cells to work with to do experiments. And that led to the development of cell culture medium with the ultimate goal being to just get the cells to grow and to grow rapidly. But the problem is the desire to get those cells to grow rapidly led to artificial conditions, because under normal conditions, cells don't grow, and double once a day like most cell types do in culture. And the result of that rapid growth can be a change in the phenotype and lead to phenotypic drift. So how can you control this? So drift can be minimized simply by limiting the number of population doublings, or passage number, that you use cells. And another way to deal with phenotypic drift is to standardize the conditions to subculture cells. And an example, I've mentioned this before, that for instance, if you're using an anchorage-dependent cell line and you're doing a trypsinization procedure to subculture the cells, sometimes you take that flask that you've trypsinized, and if you look at it under a microscope, you might still see a few cells remaining in that flask. So by doing that type of a trypsinization procedure, what you're actually doing is selecting for the cells that are more easily removed from the flask. So it's just one example of selecting for a subpopulation of cells that can lead to phenotypic drift. Okay, now this slide is actually an example of a resource. Now, at the end of my slide deck, I have an appendix that contains numerous other slides with resources. And I put this one in here just as an example. Most of them have active links that if you have a PDF copy, you may have to copy it and paste it into your browser to get to it. But there are a number of resources that will be available that I won't have time to cover during the presentation today, but nonetheless, you should have access to them. So, important point for reducing phenotypic drift is to establish a standard operating procedure for handling the cells. Some of the things that you want to include in that SOP are, first of all, obtain your cells from a trusted source. And I'll address that in more detail when I talk about cell line authentication. When you get those cells, you want to establish a master and working cell banks, but that really depends on the size of your project and how long you will plan to use those cells. It may be more convenient or simply less expensive to simply repurchase vials of authenticated cells from that reputable source. Another thing you want to do is to perform routine testing for contamination. Bacteria and yeast contamination, those are easy to see, or smelly. You can smell sometimes the yeast. And easy to deal with. You just bleach them and get rid of it. Mycoplasma is a little more difficult to detect, but there are commercial kits available for you to test for mycoplasma contamination. Other eukaryotic cells, cell line authentication. That's an issue that I'm going to speak about in the next section. That can be a problem, and it can be difficult to recognize without doing testing. And one of the things you can do is to always use your controls to confirm the performance of your cell line, that it's what it is expected. Another thing to include in your SOP is to culture at consistent cell density, and lengths of time. And I'll show you an example of that later. So using consistent culture conditions. Don't assume that all of the flasks of a stock culture of cells in your incubator are all the same. The cell density and the time since the last passage can affect the responsiveness of those cells to different assay conditions. I'll also show you an example of monitoring the energy sources or metabolites present in your culture medium, and you should know what the situation is, depending on the character or the programs, so to speak, that you're using to culture your cells. This is an example taken from a paper several years ago, and basically what it shows is an example of culturing cells at different densities in a stock flask, and then setting up parallel assays and assay plates where the cells were all seated at the same density, and the incubation was done, I think, in this case for 3 days, and then standard viability or cytotoxicity or apoptosis assays were done on those different populations of cells. And not to go into the detail of this dose response experiment, but just to say that you can observe different responsiveness of the cells depending on the history of the stock culture that happened, 3 days ago when you first set up the assay. So it's important to consistently treat your stock cultures that you're using to set up these assays. This is an example of, I don't know if you guys have something like this, but we have this email address called science. And occasionally you'll see this happen. Down here you'll say, does anyone have a spare flask of MCF7 cells I can use to set up an experiment today? And then a little later, you'll see a response to this. Someone says, yeah, I have some that I spit last Friday. I think they'll be okay. Okay, this message was sent on Wednesday. So, Friday to Wednesday is, what, 5 days. Which flask do you think they got? Okay, this one looks kind of nasty, but that's probably what they were given to set up their experiment. Another thing that happens is changing the level of energy source in culture medium over time. And sometimes it's difficult to see. This is an example of an experiment that was set up where A549 cells were cultured in a 96-well plate at 15,000 cells per well. And then assays were done to measure glucose, lactate, glutamine, and glutamate. Also, I should point out that the dotted line here represents time 0. But what you can see is, after 8 hours or 24 hours, not much change, but 48 hours after 2 days of culture, essentially all the glucose is gone from that culture medium. And that might not be easy to see, or it might go unnoticed depending on, the situation. So glucose gets utilized through glycolysis, generates lactate. Lactate or lactic acid in general is what causes the pH to change. And that's probably what caused that flask to show up yellow by the phenol red, detecting the acid. Another example, glutamine can be used up and generate glutamate. So the take-home message here is understand what's going on in your culture and monitor metabolites to make sure that you're not running out of energy. Because I can tell you right now that if you have no glucose left in a culture, it's likely that the responsiveness of those cells to a treatment is going to be different than having adequate energy source. Okay, one of the options you have is to be able to use cryopreserved cells for setting up an assay. So there are several examples where you can actually use cryopreserved cells, thaw out a vial, dispense them directly into an assay plate, and run an assay. And if you can validate that the assay performs and functions properly to give the information you're seeking, you can use frozen stocks of cells to set up your assay. And basically what that does is it eliminates the need to culture cells ahead of time. So instead of going to an incubator and getting a flask of cells to use to set up an experiment, you can simply thaw a vial and dispense it and run the assay directly. And what you can do is you can freeze down very large batches of these frozen vials of cells and that can serve as a more consistent reagent, so to speak, that can be QC performance tested. And so you can validate that that frozen vial of cells is more consistent than dealing with the variability of cell culture. And there are several options. There's vendors that actually sell you cells that you can just basically thaw and use and throw them away. So some of the advantages of using cryopreserved cells is improved consistency. So once frozen, the same cell batch can be used over and over for a long period of time. Also gives you some increased flexibility. You don't have to worry about scheduling to have your cell culture team have enough cells available to set up a screening assay. Instead, you can just go to the freezer and get vials of cells to set up the experiment. And also, depending on your particular situation, you can actually do this less expensively than having someone maintain cell lines and culture that are available for a screening campaign. And I should actually acknowledge Oliver Waymeyer for giving me some slides to use here, just as a pictorial example of the types of things that you can do with using this cryopreserved approach. So, you can scale large expansion of cells, and you can use them to create hundreds and hundreds of vials of frozen cells. Typically one of the most important things is the rate of freezing and the rate of thawing the cells to get viable responsive cells to use in these experiments. You can automatically dispense the cells to create these frozen vials for future use. And again, some of the advantages I already mentioned, but it's easy to simply go to your liquid nitrogen tank and get a vial of cells rather than worry about culturing them and having everything ready when you need to use cells to set up an experiment. However, I should say that doing it right does take considerable investment. If you are going to follow this approach, there's a lot of things to consider. There's additional information in the appendix to go over some of this stuff, but storage of the seed stocks. When you obtain a cell line, you want to probably set up a master cell bank and working cell banks. You have to validate different production lots of cells are all going to be the same. The process that you go through to freeze these cells has to be controlled. So there's a lot that goes into this. However, the advantage is you have a QC tested source of cells to set up subsequent assays. And in case you have a flood or a hurricane, you probably want to store these in multiple locations to avoid a catastrophe. Okay, I'm going to switch gears a little bit now and talk about another source of variability, and that is cell contamination and how you deal with that by authenticating cells that you use in your assays. So I use this little cartoon. They say everything was going along fine until they discovered their HeLa cell line expressed Y chromosome markers. Okay, that could be a disaster. It could be everyone in the lab is using a cell line that's contaminated. These guys are going under stress, and this guy here, he's actually going back to his desk to write a retraction to his Nature paper because he found out that his cell line was contaminated. Okay, it turns out that this problem was recognized a long time ago. Over 50 years ago, there are publications that demonstrate that people were already aware of this type of a problem. And in one particular case, Stanley published this paper, and he evaluated 20 different samples, and 18 of them, or 90% of them, were HeLa cells. Okay, so HeLa cells, easy to grow, first cell line that was used, and people were not that familiar with good cell culture practices, and it was easy to have a rapidly growing HeLa cell get contaminated into another cell line that you may be working with and take over that culture. So cell line authentication. So why is this a problem? there are numerous people who have done analysis of cells that people use and the behavior that people do to select and use cell lines. And one of the things they found is that about a third of researchers get their cells from, a colleague, someone down the hall. And oftentimes there's not documentation to go along with them that has demonstrated that it's an authentic cell line. Also, these are slightly older data, but nonetheless, only about 10% of authors authenticate their cell lines. Now, I actually speculate that this percent has gone up quite a bit recently because of the requirement of some journals to have you address this authentication issue. Also, there are numerous published reports that demonstrate that in some cases, 18 or in some cases as high as 36% of the cell lines that are being used are actually misidentified. They're not what the people think they are. That can be a huge issue, okay? And it's sort of a problem that some people are ignoring by not setting up procedures to authenticate their. I added this publication in the presentation. This was a study that was recently done in China. They looked at 22 different institutes and they evaluated 278 different cell lines. 128 of them, or 46% of those cell lines, were not what they thought they were. So again, this is a huge problem, but it's easy to address. Another example of a publication, this had to do with this metastatic breast cancer cell line that was isolated originally at MD Anderson back in the 70s. And this paper by Christopher Korch describes sort of the chronology of the development of the cell line. And what happened was it became contaminated with a melanoma cell line. And over the years, there were publications that identified this cell line as being contaminated. This, by the way, was actually lines that was added to the NCI 60 list. But there are publications demonstrating that this cell line was contaminated, yet people continued to use it. And what this paper describes is sort of the history and how many different times the cell line had been used and published on, even though it existed in the literature identified as a problem. So what is being done about cell line authentication? Well, certainly there's increased awareness in the scientific literature. There's more and more people realizing that this is the problem and setting up procedures in their lab to deal with this. The NIH has published notices encouraging scientists to authenticate their cell lines. And this is now in the grant application process, and then there's more and more pressure on scientists to follow appropriate practices. Many of the journals are now actually requiring people to address this issue as they submit manuscripts. And I don't have time to go through them now, but these notices and the list of journals that mention this, there's information about those in the appendix to this presentation. Also, there are numerous testing services or core labs that you can go to have your cell line authenticated. There's also a list of core labs in the appendix. It's called the International Cell Line Authentication Committee, or ICLAC. You'll hear people talk about this acronym. And over the years, they've been active in this area. And in 2012, they published this authentication of human cell line standardization of STR profiling. The good news is that this is actually now in the process of being updated. And Christopher, the person I mentioned with that breast cancer cell line publication, he has actually revised this, and it's out for review right now. So this, you can anticipate a new version of this coming probably in 2021. Also, in the assay guidance manual, there's an existing chapter that was written by Yvonne Reed from ATCC. We're also planning to update that chapter to contain the updated information from this ANSI national standard. So several additional resources, as I mentioned, are included in the appendix, along with lists of many different publications that address this issue, which you might be interested in seeing. So how does authentication work? Well, it's really pretty simple. There's a database of misidentified cell lines. The first thing you want to do is to check that database to see if your cell line or the cell line that you're using is already on that. I included a link here. I won't take the time to go through it, but this links to an Excel spreadsheet that shows cases where cells have been misidentified. And if I remember correctly, that spreadsheet goes on for 28 pages. So hundreds of rows identifying or mentioning cells that have been found to be contaminated. But how do you do this? How do you get cells authenticated? Well, what you can do is simply take a sample of the cells or extracted DNA and simply send them out to a core facility to have it tested. ATCC does this. There's also a number of facilities and universities that will do this. Send them the sample, and then they'll do the profile and send you back the information. And they typically also, provide you with information about whether or not it's considered to be contaminated. So in some cases, you can order this to be done. And companies like ATCC, I think they still just like mail you a card for you to pipette a sample of cells on, and then you simply mail it into them, and they'll run the STR profile. So it's relatively simple to do, and it's not that expensive to do either. So big bold letters here. Investing in cell line authentication is good science, and developing an authentication program should be part of your good cell culture practices SOP. If you take anything home from this presentation, if you weren't already aware of this problem, I want to make you aware of it and convince you that it's worth your effort to develop this as part of your SOP. Okay, another topic I want to address as far as addressing the reproducibility of cell-based assays is using cell number as an internal control. And what do I mean by that? So confirming cell number as a normalizing control will help you improve the reproducibility of your cell-based assays. So reproducible data really depends on having a consistent number of cells in each well. So the issue is that the number of cells can change during the course of an experiment, especially if you're incubating for longer periods of time. 2 to 3 to 4. I've even seen people go, 7 days in a culture well without changing medium. So knowing the number of cells at the end of the experiment can be used to understand proliferation or death that occurs during the course of incubation. It can be used to normalize the data. It can also be used to identify pipetting errors or edge effects that are involved with setting up the assay plates. So I should also extend this, not only knowing the number of cells at the end of the experiment, but knowing how that cell number may change during the course of the experiment is important. And another good recommendation for a control is to know the number of viable cells or total cells at the beginning of the experiment as well. So, and whether or not that value changes over time. Some of the situations that can lead to changing in the cell number. A classic example is someone who's doing a reporter gene assay. And if you're doing something like a luciferase reporter gene assay and you see a decrease in signal, is that a specific downregulation of that gene, or are you simply killing the cells? So by doing a viable cell number count, you can figure out which case it is. So if the compound causes cell proliferation or stops division, maybe it's cytostatic, or it can alter metabolism or induce cell stress, those are all situations that can apparently change cell number. And the one I should point out here is altered metabolism. And just beware that some of the assays that you do to normalize for, especially for viable cell number, what they're actually doing is measuring some metabolic event. So just be aware that if you're changing the metabolism of cells, the type of data that you get from some of these cell viability assays will also, it may not correspond with the total number of cells. Also, what if there was a dispensing error that resulted in fewer cells in the well to start with? So that's another example of an error that you can pick up by doing a total cell number characterization of the assay plate, even at time 0, to validate the process itself. So differences in cell number, how do you detect them? There are examples of assay technologies that are available to multiplex the measurement of live, dead, or total cells, and you can use that data to normalize the experiment, normalize the data, and improve the reproducibility. There are many options available to measure cell number that are compatible with other assay chemistries. And when I say that, what I mean is that you can use them in a multiplex format. Also, there are real-time assay methods available that provide you a lot of flexibility and the opportunity to multiplex. And I'll show you examples of measuring viable cells or dead cells or total cells in real time. And without taking a lot of extra time, I should point out that there are chapters in the assay guidance manual dealing with these different topics. There's a chapter on the cell viability assays that goes through more of the details of the assay chemistry. It identifies the types of things that interfere with those assay chemistries and shows you the advantages and disadvantages of using the various assay methods. So this is an example of how a real-time viability assay works. And the way that this assay works is there's an assay reagent that you add directly to cells and culture, and that reagent contains a pro-substrate for a shrimp luciferase and the recombinant luciferase itself. So the recombinant luciferase and the pro-substrate is added directly to cells and culture. The pro-substrate penetrates live cells and it gets converted into a substrate for luciferase. So as long as you have a viable population of cells there, you will get a glow signal. As soon as the cells die, they lose the ability to convert the pro-substrate and there's no signal. So it's only the live cells that give you a signal. And this signal, by the way, or the conversion of the pro-substrate to the substrate is a reduction reaction. So this works similar to a tetrazolium assay, like the tetrazolium would be reduced to the formazan. So in this case, you're simply reducing a pro-substrate to a substrate. So as long as you're aware of this mechanism, it's sort of an indicator of whether or not you should be aware of things like reducing agents to potentially interfere with this type of a measurement. So the example of using a real-time assay, in this case, this is a 3-day experiment where a dose response of a model toxin was set up. And what you can see is by recording luminescence over time, and the way this experiment was done, by the way, the plate was set up, the assay reagent was added once at time 0, and then it was put in one of these plate readers that has an environmental chamber, and it was programmed to record luminescence every hour. So you can see the vehicle control here on the top, or the lowest concentrations of toxin. You get more and more luminescence over this 3-day period. And the wells that has the highest concentration of toxin, you see a decrease in the luminescence signal over time. You're killing those cells. So you can do this in real time and monitor, what's going on in that culture. Okay. There's also a chapter in the assay guidance manual about measuring dead cells. And I'll describe one of the assays that you can do in real time using this approach. There are fluorescent compounds that are typically referred to as vital dyes. And the way that they work is you can add a fluorescent dye to cells in culture, and there are examples of dyes that are not permeable to live cells, but will only penetrate cells that have lost membrane integrity, and then they'll bind to the DNA and stain the DNA, some color. In this case, the example is it stains it green. So this is an example of using this dye. This is a dye called CellTox Screen, and it's done over a 3-day period. So the way that this experiment was set up is, cells were cultured and in the presence of a vehicle control, concentrations of toxin. In this case, this was a terfenadine that was added. And then in this case, fluorescence was recorded repeatedly over this 3-day period. And what you can see is with the vehicle control or the lowest concentration of terfenadine, there's no change in fluorescence over this 3-day period. But with the intermediate concentration, you'll see a gradual increase in fluorescence, going out, starting, almost immediately. But what that's telling you is you're accumulating dead cells in that culture, okay? So that's what's going on by seeing increase in fluorescence. And with the highest concentration of toxin, you see you pretty much kill the cells right away. But also important is that this fluorescent signal is relatively stable over that 3-day period, which is, by the way, one of the problems of using something similar. In some cases, the release of lactate dehydrogenase as a marker of dead cells can be problematic because in some cases the LDH may be less stable and you'll see a decrease in signal after 3 days in culture. So how can you use this type of real-time data? So as an example, when you're monitoring a plate that has a library of compounds across it, what you can do is do sort of a real-time monitoring of individual wells to determine which compounds are resulting in cytotoxicity. And I call this the cherry picking example. So because the real-time reagents themselves do not kill the cells, there's a lot of different opportunities for multiplexing. And you can, as I mentioned already, monitor the kinetics of cell death over time. So individual wells that show changes can be cherry-picked and set up secondary assays. And this is an example of the type of multiplexing orthogonal data that you can see by running a combination of that DNA binding dye to measure dead cells, along with that luminescent real-time assay to measure live cells. Now, to be honest, sometimes I use this graph as an example of what not to do, of showing too much data on a slide, but you can see the general trend of the increase in fluorescence over time with higher concentrations of toxin, and that corresponds to the decrease in the viability marker. So an orthogonal method of looking at the increase in dead cells corresponding to the decrease in live cells. So this is just a flow diagram depicting how this cherry picking type of experiment would work. You can screen a panel for cytotoxicity, for instance, and when you see an individual well light up or start to light up, what that's telling you is some of the cells in that well are dying, okay? Other members of that population of cells are probably undergoing earlier phases of cell stress or the stress response, and you'll find by analyzing that you'll be able to identify which signaling pathways are contributing to the early phases of that death process. So just an example of real-time monitoring of, in this case, dead cells that you can multiplex with the detection of some other parameter. So the general summary and take-home messages that I want to convey by this using cells as reagents presentation is, first of all, seek to understand the nature of cells in vitro. As I mentioned jokingly, they don't think for themselves, so you have to do the thinking for them. Be aware of the problem of phenotypic drift and the things you can do to avoid it. And probably the most important message here is that you should establish a standard operating procedure for consistent handling and culturing of cells and have everyone in the lab follow that procedure. One of the things you can do to avoid some of the variation, if it will work for you, is to consider using frozen cells with a known set of performance specs. And It can remove some variability that depends on having a consistent source from stock cultures. Also, as I hope I've been able to convince you, it's worth your effort to authenticate the cell lines. And when I say at appropriate times, there are recommendations from that ANSI standard of how frequently you should do that. If you're using good cell culture handling procedures, you can decide yourself how frequently you want to authenticate your cells. Also, there's the opportunity to normalize to cell number to identify problems and reduce variability. Use the ability to multiplex assays, to find out what's going on in that culture over time. And some of the advantages of using real-time assays, hopefully that'll improve the efficiency and enable multiplexing as a control much more easily than using something like an ATP assay, which is an endpoint assay that would destroy all of the cells. So with that, I'll hopefully stop here. And if there are questions beyond this presentation, you can feel free to email me. And also I want to mention that there is an appendix of additional resources of several slides at the end of this presentation. And hopefully Serene will describe how you can get access to those slides. If not, you can send me a message and I can provide them to you. Okay, Serene, do we have questions?
Yes, thank you for the wonderful presentation, Terry. We do have one question, and this is referring back to the assay-ready cells. The question is, doesn't toning cells stress cells, though, so might not get physiological results if using right after tow?
Yes, great question. Freezing cells, thawing cells may cause stress. Basically, anything you do to cells in culture could potentially interfere with subsequent assays. That includes, adding a vehicle, adding more PBS. That could potentially affect the cells as well. Whatever you do to them, you should validate whether or not it has an effect on the subsequent assay. As long as you're using, if you're using frozen cells, as long as you can validate that the assay you're using is what's called fit for purpose, then you should be okay. As long as you're able to answer the experimental question you're asking with those frozen cells, you should be able to do it. Because keep in mind that cells in vitro are simply a model. And the components you improve or include in that model being the cell culture and the assay for interrogation should be considered as part of that total model. And if it works for your purpose, if there's a slight interference, that may not be too much of an issue.
All right, thank you, Terry. We do have a couple of more questions coming in for you. So one of them is, are the connectivity map, CMAP, and TOX21 also updated and authenticated? And it follows by, how do you prevent glucose and other sugars from being taken up by cancer cells, Warburg effect?
Okay, I'm not sure about updating or whether or not the TOX21 database information is updated. I would have to defer to someone else. I think Meng Hong, who's going to report or present later, would probably more effectively answer that question. And the question about glucose, would you repeat that part of it, please?
Yes, absolutely. So how do you prevent glucose and other sugars from being taken up by cancer cells? Warburg effect.
Well, I'm not sure there's an easy way to prevent cells from taking up glucose. I think just by assaying the culture medium and being aware of the changes, is certainly informative, but I'm not sure that there's a way to stop cells from taking up glucose, and that would probably, change the biology of the assay system.
Thank you, Terry. There's one more question here from Dr. Samarjit Patnaik on the panel. How easy it is to set up STR profiling in every lab?
Well, if you're a biologist like me, it would be difficult because what you need is a capillary electrophoresis instrument. The details of setting up the assay will be included in that new standard, in the new ANSI standard. I can tell you that depending on where you are in the world, there are going to be, I think, different requirements for as far as how many markers are used to be considered, like a valid call on whether or not something's contaminated. I know that's certainly the case for human identification, which uses that same or actually an extended set of those STR markers.
Thank you, Terry. You have more questions coming in, but it's 11:30 right now, so I do not want to erode into Dr. Joe Traska's time. So do you mind just answering those questions yourself?
Yes, if those questions are sent to me, I can answer them. I also should point out that some of this will be extended during the one of the roundtable discussions a little later over the lunch period, the cell-based assay. So I'll be included in that as well.
Okay, so it's in the question and answer chat box, Terry. So you should be able to look at the chat box and answer those questions.
Okay, I'll try to do that.
All right. Thank you very much, Terry.
Thank you.